Welcome everyone, Mike Fave here. So I've been seeing a lot of conversation, at least in the spheres that I've been in, talking about heart disease and LDL cholesterol and basically discussing LDL cholesterol as a causative factor. And so with that, and also with my time in the hospital, I kind of wanted to put together a presentation to discuss what's actually going on inside the artery, not only coronary arteries, but the large arteries in general in terms of heart disease. Now, I want to put some context for this, particularly from like a metabolic or bioenergetic point of view. Cells need oxygen and nutrients in order to function at their metabolic peak, in order to effectively oxidize either carbohydrate or fat appropriately. So, in situations where there's not enough supply of whatever the substrate is, carbohydrate or fat, and also oxygen, the cell can't produce any energy, and then you get issues where the cells essentially die. And it doesn't really take that long for the cells to die. So circulatory health is extremely important for metabolic health because it allows for the distribution of nutrients. So if your circulatory system is in any way compromised, then your ability to produce energy is compromised directly as well. So maintaining adequate circulatory health is extremely important in metabolic health. And so I want to kind of dig into here, what are the actual mechanisms, in this first video at least, what are the mechanisms that lead to the formation of plaques and damage to the arteries? And when subsequent videos, my goal is to go through and talk about the different things impacting this process. So the title of this current video, or this whole series, is Coronary Artery Disease and Friends. And that's because there's quite a few factors that directly impact coronary artery disease. So you have some basic ones just to mention off the top that I'm going to for sure go over are going to be endotoxin. It's going to be vitamin C. It's going to be polyunsaturated fatty acids. There's going to be some talk about thyroid. We can talk about the renin-angiotensin-aldosterone system. We can talk about glucocorticoids and stress hormones in the sympathetic nervous system. So we can talk about all of these different factors and discuss. And another one also would be iron, and discuss them in the context of damage to the vascular wall. So it's not as simple as high cholesterol heart disease. There's more to the picture going on. So we have multiple factors underlying this process that leads to this damage to the vascular wall. And then we have a reparative process that can either lead to plaque formation or to healing of the artery. So let's jump in and we'll start to just see initially here, what is the actual process that occurs for the formation of plaques inside the arteries. Now I listed coronary artery disease here, but a lot of the major vessels are directly affected by this, including the aorta, some of the femoral arteries, etc. So first video here is going to be the mechanisms of atherosclerosis. Now, the atherosclerosis generally occurs at very specific areas in the body. Usually you're not seeing it down in these lower vessels or in the more distal vessels that you have in the arms and whatnot. Usually it's around these higher pressure vessels around the heart and these larger higher pressure vessels, the abdominal aorta, iliofemoral arteries, coronary arteries, carotid arteries. So these major centralized vessels. So we have a quote here. This quote says, "Atherosclerosis is a multifocal disease that attacks reproducible regions of the arterial tree. Sites with low or oscillatory endothelial shear stress located near branch points and along inner curvatures are most susceptible, and the abdominal aorta, coronary arteries, iliofemoral arteries, and carotid bifurcations are typically the most affected." So basically what they're discussing, and we're going to get into what is oscillatory endothelial shear stress, and we'll talk about why branch points and curvatures are important to the formation of that type of stress on the artery and what that stress does in terms of the formation of plaques and whatnot. So basically, all we're seeing here is just some basic graphic showing the major areas where plaques develop in humans, and it tends to be these more centralized regions around the heart and just coming out of the heart, and then the larger vessels moving to the lower body and then also to the brain. And something to keep in mind here, something interesting, is that men tend to have a bit more atherosclerosis than women do, as you can see based on the percentages. So I'm not going to dig into differences between men and women at the moment or in this video specifically, but we can get into that in other videos. So let's zoom in a little bit and we'll talk about what's going on at the aorta and the carotid arteries. So we have another quote here, and I have the most common sites of atherosclerosis are where blood flow is divided. So this anatomic relationship is no coincidence because many lesions occur near flow dividers, as there's ample evidence to suggest that atherosclerotic lesion size and vulnerability can be manipulated by changing patterns of blood flow. So something that's interesting is just the blood flow itself puts stress on the arterial structures. And that's really important to keep in mind, that the arteries aren't just these old pipes that over time degrade. They're constantly repairing themselves because the blood flow itself has, depending on how the blood is flowing specifically, has a somewhat damaging effect on the arterial structure. So basically what they've found in research is that the certain areas of the artery are more likely to be damaged by these changes in blood flow, and it's usually around curvatures, as you can see here in the aorta, and or like the carotid arteries where you have these branch dividers. And because the division and the curvature lead to changes in blood flow, and that change in blood flow leads to increased stress on these specific regions of the arteries. So there's specific areas that generally form plaques more so than others. And you can see that there's like known, that's why you can, when you talk to your cardiologist, you can say, oh, this area is known as the widowmaker, because there's generally certain spots in the heart and the major vessels that will get certain arteries. And when those areas get occluded, it's to large portions of tissue. And so for like with I think the LAD is the widowmaker, if I remember correctly. So when that artery gets blocked, a large portion of heart tissue dies, and essentially you die because you can't move enough blood to your tissues. And actually, there's a chance that I'm going to go in a video and discuss the like theories around blood flow and the heart as a pump and whatnot. So perhaps we'll get into that, but I want to kind of talk about the mechanisms of atherosclerosis first. So next slide here, this is where we talk specifically about blood flow. So we have there's two types of blood flow, laminar blood flow versus turbulent blood flow. So we'll get into this quote here, which will describe it for us. So what I have here, turbulent blood flow as an initiator of atherosclerosis. So the quote starts, in the arteries, the shear stress depends on the phase of the cardiac cycle, increasing during systole, so that's when the heart is contracting, and decreasing during diastole, so when it's relaxing. The geometry of the vessels, such as bends and bifurcations, also affect the nature and values of the shear stress. In the straight parts of the arterial tree, when blood flow is laminar, so what we can see over here, this is the laminar blood flow. The blood is moving in a singular direction. Shear stress is high and directional. While laminar blood flow is considered physiologic, the appearance of turbulence is seen as an important initiating factor for atherosclerosis. The transition to turbulence is associated with curvature, stenosis, branching of the arteries, and is accompanied by the formation of disordered chaotic flows. Such impaired blood flow patterns are characterized by an irregular distribution of small shear stresses. So what we're going to see in this next picture, but specifically like what's going on with the endothelial cells, but you have two different types of blood flow. So in a kind of straight vessel, a non-curved vessel, or an area that doesn't have a branch point as we see over here, the blood is flowing in a straight pattern. So there's no real, like, the stress is all, there is stress along the arterial wall, but the signaling of the blood moving in this particular pattern actually signals these pro-survival genes inside the endothelial cells that keeps them in a good shape, essentially, and allows them to function appropriately. However, when you get these bifurcations in vessels, or you have curvatures as we saw in the aorta just a second ago here, what winds up happening is you develop these areas where, as the laminar blood flow shifts, you get these small little eddies, or like you would see in a stream, kind of like if there's an offshoot in the stream, the water kind of flows and pools in an irregular, turbulent manner, as opposed to moving straight in a directional manner. So what you see here is this lack of laminar flow and this turbulent flow changes some of the gene expression on the endothelial cells, and actually leads to an alteration in the amount of shear stress, and then can lead to some breakdown of the cells in these particular areas. So the turbulent blood flow is not ideal. The laminar blood flow is ideal. And again, areas where the vessels bifurcate or they split, or where there's curvature, or if there is stenosis in the vessels, which usually happens later on after there's significant damage to the vessel, that can lead to further formation of these eddies in this turbulent blood flow, and that triggers an inflammatory effect on the vessel wall directly. So just the flow changes in blood flow itself can affect the vessel wall. Now other things can as well. For example, oxidized fatty acids, endotoxin, high amounts of iron, inflammatory cytokines and mediators. All of those things can be injurious to the vessel, but just something to keep in mind at the moment. Why are we seeing the most amount of formation of plaques and the centralized arteries around the heart and the carotid arteries in the aorta is largely because of their alterations in blood flow, branching of vessels, etc. And also these are the higher pressure systems compared to the more distal vessels. So next thing we get into, and this is kind of talking about or showing a close-up effect of the turbulent or oscillatory blood flow, and then I'm not going to go into the specific mechanisms here, but basically what this discusses is that this laminar shear stress, where the blood is moving in this kind of linear direction, triggers these different pro-survival anti-inflammatory signaling pathways compared to the oscillatory turbulent blood flow, and then that changes what's going on with the expression of the endothelial cells, in the artery, and then that can lead to the formation of plaques, or as you see here, those are the areas that are more likely to develop plaques. So what we have here, initially I have turbulent blood flow induces pro-atherogenic genes involving extracellular matrix proteins. Now, the extracellular matrix proteins, we're going to hone on in just a sec. They're extremely important. So what we see is essential hemodynamic elements underlying the pathology of atherosclerosis involve shear stress, oscillatory or bidirectional flow, and local eddies and or boundary layer separation. The influence of hemodynamic forces along with changes in proteoglycan composition is one of the primary contributors to early lesion development because mechanical strain is thought to influence a variety of pro-atherogenic genes, in particular those involved in the production and or processing of extracellular matrix proteins. So that turbulent blood flow, that oscillatory blood flow, or the non-laminar blood flow, this type of blood flow here that you find at the curves or at bifurcations or even at areas of stenosis or narrowing, leads to the turn on of these inflammatory sets of genes and signaling pathways that upregulates extracellular matrix protein production and and alters proteoglycan structure. So extracellular matrix proteins and proteoglycans are components that are structural components within the artery, within the arterial wall, that are kind of like have a collagenous structure to some extent, and I guess that's the easiest way to think about them, but it's kind of like the plaster board of if the artery was a wall, you kind of have the walls are formed by plasterboard. So inside the wall there's different studs that provide the structural components, and then there's what you call the insulation panels. So like I don't know if everyone's familiar with this, but there's kind of like these insulation panels inside the walls of your house, and they're made out of what's it called? Not styrofoam, fiberglass. So you have the fiberglass insulation in there. So you can think of the extracellular matrix proteins and the proteoglycans as kind of similar to that fiberglass insulation that you would have in an exterior wall of the house. So the artery has these extracellular matrix proteins and proteoglycans inside the wall. And so you can see here inside the wall here. We'll get better pictures as we go along. And so the strain from this oscillatory flow or this turbulent flow upregulates the production of these proteins. And this is going to be extremely important because this is what initiates the LDL, the cholesterol binding into the artery. So we'll see that in just a second. So first things first, and again, this is an extension of what I was saying before. So we see increased extracellular matrix leads to arterial intimal thickening. So quote here, in human vessels, proteoglycans are mainly found in areas with intimal hyperplasia. Intimal hyperplasia or diffuse intimal thickening is the thickening of the innermost vessel wall layer, the intima, due to accumulation of vascular smooth muscle cells and extracellular matrix. So here we have a dissection image of an artery, and this is a healthy vessel versus an unhealthy vessel. and basically it breaks down the structures. So with the artery, you have three layers here. There's actually more like 4 layers. So they're showing 3. So you have the intimal layer. Here you see the endothelial cells lining, and then there's kind of a layer in between the endothelial cell lining to some extent in the media. The media is the muscle layer, the smooth muscle layer, and then the adventitia is like a connective tissue layer. Now, when you have damage to any type of irritating effect or damage to an artery, it could be the blood flow, turbulent blood flow, as I was discussing before. It could be endotoxemia. It could be oxidized lipids. It could be those inflammatory cytokines and cellular mediators. All of those can upregulate these processes. But what you start to see is when the artery becomes injured, it kind of forms a callus to some extent. It starts ramping up the formation of these extracellular matrix proteins. And what you see is this intimal thickening or intimal hyperplasia. So this layer right below the endothelial cells starts to get thickened and starts to increase these sticky proteins, these extracellular matrix proteins, kind of that insulation to some extent in between this nice endothelial layer, this is the lumen of the vessel, the endothelial layer, and then the muscular layer, the media. And then it kind of merges with the media and this intimal layer. And so they call this the neointima. So basically the vessel becomes thickened. It kind of forms this callus to some extent. This is more of an analogy. It's not perfect, but it gives a good idea. Like if you get a cut or injury on your hand, the skin's kind of thickens up a little bit. And so you see that inside the artery as well. Now, basically what we're going to discuss here is that any irritant or damage to an artery leads to thickening of this intimal layer, and it's via, again, that increase in extracellular matrix proteins. And again, I'm harping on the extracellular matrix proteins and the intimal layer because this is where the cholesterol essentially binds into, and we're going to see that in just a few minutes here. So what we see is neointimal thickening as a response to injury. So neointimal formation, however, occurs in all arteries as a response to a wide variety of injuries, including radiation, application of turpentine to the adventitia, wrapping the vessel, and electrical stimulation, as well as mechanical injuries, including placement of a suture, scratching with a probe, or dilation of the common carotid artery with an embolectomy balloon catheter. These changes are even seen in transplanted veins that undergo arterialization. Put another way, neointimal formation is a characteristic of responses to injury in the smooth muscle-rich vessel wall. So again, I have another image here. You have a healthy artery, you have a diseased artery, and what you can see is the different layers. So those media layers, that muscular layer, that adventitia layer is that connective tissue layer. You have the lumen of the artery, and then you have this intimal layer, the closest layer to the lumen, and there's a bunch of endothelial cells that line this intimal layer. Now, when you damage the artery, you irritate it in any type of way, changes in blood flow. Here they talk about a bunch of experimental damage that they've induced by different methods. What you find is that this intimal layer gets thick and it increases with those extracellular matrix proteins. The purpose that I had for this slide was to just give you guys some proof to show that any type of irritation to the artery leads to the formation of the thickening of the arterial structure, where the intima gets thick, and then you can see some of the media become thick. This kind of the barrier between these two kind of gets disrupted as the damage progresses. So again, now what we're seeing is that just as a quick recap, the most likely areas of the vasculature to become damaged are the higher pressure arteries around the heart, around the aorta, and the larger vessels that are moving towards the legs, which is in the femoral arteries, and then the carotid arteries moving towards the brain. And what characterizes some of the damage is the curvature in those arteries, as well as the branching off in those arteries, and they are the higher pressure systems as well. The other thing that we're seeing here is that when those arteries have any type of irritation or damage, they thicken up this intimal layer, and they do so by increasing extracellular matrix proteins, which are kind of like this sticky, insulating proteins to some extent that kind of build up in this middle or this inner layer of the artery behind the endothelial cells in the intimal region. So that's a general response we're seeing for arteries. Now, we're going to start to talk here about atherosclerosis and then the formation of, or the trapping of lipids. This is where it becomes really important, because you have that thickening of the matrix first, and when that intimal layer with the extracellular matrix, and then that sets the stage, basically creating a nice net to capture these lipids. So what we see here is arterial intimal thickening, a reservoir for lipoproteins and lipids. The role of intimal hyperplasia as an initiator of atherosclerosis is supported by a recent autopsy study from Nakashima and colleagues. They analyzed lipid deposition and macrophage infiltration in coronary arteries from children and young adults less than 40 years old who had died from non-cardiovascular events. The degree of atherosclerosis was classified according to the amount of lipid deposition in the intimal lesions of the arteries. So again, in this intimal layer, and we can see that here, they found that extracellular lipids are trapped in the diffused intimal thickening that we can see over here. At low grades of atherosclerosis, this lipid accumulation is present in the absence of macrophages. So macrophages are immune cells, and macrophages appear in lesions with increased lipid deposition. So we're going to talk about this. Thus, diffuse intimal thickening likely acts as a reservoir for lipoproteins, and macrophage infiltration in infiltration is subsequent to lipid deposition. So we have kind of these steps: damage to the artery, extracellular matrix accruing in the intimal layer, you form this thick neointimal layer, and now this thick neointimal layer acts as a net or a reservoir to start trapping these lipoproteins like LDL. So something to keep in mind here is it's not just this high LDL is just moving into the arterial wall, because you have the LDL moving into the arterial wall in areas that have the increased extracellular matrix due to thickening from some type of irritation. And again, multiple forms of irritation can cause that: inflammatory signaling, endotoxemia, excess iron, oxidation from polyunsaturated fatty acids, turbulent blood flow, etc. There's a multitude of things that could lead to this irritation to the artery. Again, these are relatively delicate vessels. So now what we're going to talk through is, what does this whole process look like in terms of the irritation to the artery, lipoproteins coming in, binding to the extracellular matrix, the lipoproteins becoming oxidized, attracting immune cells known as macrophages, and then those macrophages perpetuating this response. Because what we're going to see is this is a feed-forward response. So there was a hypothesis generated to describe this process. This hypothesis is called the response to retention hypothesis. So this slide I have here is an overview, but we're going to break down each of these steps individually. So the quote we have here says, "The response to retention hypothesis emphasizes that the interaction between the extracellular matrix molecules and lipoproteins and the modification of these two molecules are the key events in early atherogenesis. This hypothesis is not inconsistent with the chronic inflammation hypothesis, because retained and modified lipoproteins can stimulate the recruitment of inflammatory cells such as macrophages and T cells." Now, this is going to be really important, particularly this part where we talk about the retained lipoproteins stimulating inflammatory cells, because the lipoproteins, the LDL, if it's oxidized, can actually trigger the toll-like receptors, which are receptors on immune cells that even endotoxin triggers, that lead to basically an upregulation of immune response. So when the lipoproteins are filled with polyunsaturated fatty acids and you have a damaged vessel wall, they can migrate into the vessel wall and get trapped in that extracellular matrix. The macrophages, the immune cells, can sense them because they're oxidized, and those oxidized lipids are going to trigger the immune cells to come in. The immune cells gobble them up, and then, as we'll see in a few minutes, they become something called foam cells, and then they eventually die. And so there's a whole process. We're going to go through the rest of it. So first step in the response to retention hypothesis is predisposing stimuli increases the synthesis of extracellular matrix proteins. So we have a quote here: "It is further postulated that this process begins with predisposing stimuli, e.g. mechanical strain and cytokines," we discussed that enhance local synthesis of proteoglycans with high binding affinity for lipoproteins, and that atherogenic lipoproteins enter the arterial intima and are bound and retained by proteoglycans. So here we have, this is the inflammatory stimulus, this little lightning bolt. You have lipoprotein moving into the vascular wall, and then these are the proteoglycans that we'll see in a second that will bind. We'll see that on the next slide. So step 2, lipoproteins are able to bind to the increased extracellular matrix. So, quote, the interaction between lipoproteins and proteoglycans is ionic in nature and occurs between the negatively charged glycosaminoglycan side chains of proteoglycans and the positively charged residues of apolipoproteins. So, these are the apolipoproteins. This is most likely LDL. That's the more atherogenic one that people are familiar with. And then you have it moving into the wall. It's kind of triggering this increased production of these proteoglycans. Those again, that's like kind of that insulation, the fiberglass insulation inside the arterial wall that can hold these lipids now. And just so everyone's on the same page here, this is the lumen of the artery. So this is where the blood would be flowing. These are the endothelial cells. This is the intimal space, and then here's smooth muscle cells. So and then you have an LDL or a lipoprotein moving in, and then here's the proteoglycan. So just so we're all on the same page. Step number 3. Once lipoproteins bind to extracellular matrix proteins, they become oxidized and are eaten by immune cells. So eaten in quotations. Quote we have here, the hypothesis further states that lipoprotein proteoglycan complexes exhibit increased susceptibility to modifications such as oxidation and aggregation, and that these modifications lead to uptake by macrophages to form foam cells. So we're going to see the macrophages in just the next slide, but what we're seeing here is, again, lipoproteins moving across the wall. They've already increased that extracellular matrix, or the extracellular matrix is increased. It's kind of, you have this inflammatory stimuli that increases the extracellular matrix, but once the lipoproteins start to bind in and they oxidize, they can also increase those extracellular matrix proteins. And here you see them bound to the extracellular matrix proteins now. So this slide really encapsulates more of this. And so we have step 5 here. These oxidized lipoproteins further enhance the production of extracellular matrix, kind of what I was just discussing. They say furthermore, oxidized lipoproteins enhance the production of proteoglycans with a high affinity to lipoproteins. So what we're seeing here is you have the proteoglycans, you have the lipoproteins, the LDL, it's bound to the proteoglycans. Other thing, if we come down here to the key, we see that the purple balls are oxidized lipoproteins, and the oxidized lipoproteins are bound to the proteoglycans as well. These are triggering the immune cells. So we see the macrophages coming in, or I guess these would be monocytes, and when they come in, they turn into macrophages, and these macrophages would gobble up these LDL, and particularly the oxidized LDL is going to trigger inflammatory response. And you see oxidized lipoprotein here, oxidized LDL. Now, this is another view of this, so you guys can kind of see how the lipoproteins are binding to the extracellular matrix here, the proteoglycans. Those are the proteins in the extracellular matrix. So what we see is another view of lipoproteins and extracellular matrix proteins in the intimal layer of the artery. So, quote, binding of LDL to intimal proteoglycans is an important step for disease initiation, which may potentially explain the atherosclerosis proneness of adaptive intimal thickening. As the disease evolves, however, the endothelium becomes more leaky, and the expression of lipoprotein binding molecules in the plaque may further promote the ability to retain LDL. So, when the LDL is able to bind to these proteoglycans, we see that over here, it initiates the disease process, as we saw in the response to the retention hypothesis. Once the LDL is bound to the proteoglycans, or the lipoproteins are bound to the proteoglycans, they become oxidized, especially if they're rich in polyunsaturated fatty acids, particularly omega-6 polyunsaturated fatty acids, which have been characterized, and we're going to see this in PUFA, to be highly expressed inside plaques. So what we're seeing here, here's the lumen of the blood vessel. This is where the blood flows. You have the endothelial cells lining here. This would be the smooth muscle cells, and you have the neo-intimal layer here. And what you're seeing is these orange balls are the LDL. Then you see these blue kind of lines here are the proteoglycans, those extracellular matrix proteins, and they're binding up all of the LDL, the lipoproteins binding in. And then we have these immune cells kind of infiltrating the area as well. And essentially, it's this kind of feed-forward cycle, right, where the lipoproteins are binding to these extracellular matrix proteins, and they're becoming oxidized. They're triggering the immune cells to come, and you have this inflammatory response. More proteoglycans get synthesized, and then more LDL can get captured, and you start to build this fatty plaque, this fatty streak. And so we're going to see this process in just a second as we move forward. Something I want to touch on really quick before we jump any further, because it's going to become really important when we talk about vitamin C, lysine, and proline. We're going to talk about how the LDL is able to bind to the proteoglycans, and it's through these lysine residues. So we're going to see that here in this quote. So later, Camejo et al. Investigated the interactions of LDL with chondroitin 6 sulfate-rich proteoglycans. So again, proteoglycans are the proteins within the extracellular matrix by using quantitative frontal elution affinity chromatography. These results indicate that 3 peptides were responsible for interaction with these proteoglycans, that's the CSPG, and they share an excess of positive charged side chains belonging to the lysine and arginine-rich LDL receptor domain. Studies in transgenic mice expressing ApoB100 mutation have established that site B, which is this specific residue, possesses positively charged arginine and lysine residues that are essential for binding to proteoglycans. So essentially, this is the apolipoprotein on a lipoprotein on an LDL, and there's these sites on the protein that allow the LDL to bind into the extracellular matrix. So what we're going to talk about is the Linus-Pauling heart disease, Linus and Dr. Matthias Rath's model of heart disease. And basically their treatment pathway involves using proline and lysine as well as vitamin C to basically eliminate heart disease. And the proline and lysine become important because they're able to occupy these receptor sites. And by occupying the receptor sites with that and blocking the LDL and the proteoglycans from interacting with each other, you can stop that influx of these lipoproteins into the vascular wall. And then with adequate vitamin C, from their perspective, it allows the arteries to heal appropriately. Vitamin C is one of the main cofactors in collagen synthesis. So those areas that become damaged can essentially be healed with if they have an adequate supply of vitamin C and the amino acids to produce collagen. So again, we'll talk about that in a bit, but I wanted to show this here because it'll become more important as we go along, talking about the lysine binding sites on these LDL so that they are unable to bind into the vascular wall. Okay, hold on. There we go. So I just wanted to show you another picture of the LDL so you can have an idea. So the LDL here, all these little spherical structures you see, those are the fats. Those are cholesterol, phospholipids, triglycerides, cholesterol esters, etc. These are all held within this lipoprotein. The actual protein is this yellow piece you see here, and that's the same as this kind of grayed stripe that you see moving across the whole sphere here. So what you're basically seeing is the fats, the lipids here, the cholesterol, the triglycerides. Since they're fats, they're not soluble inside water. They're hydrophobic. So you need a protein to actually carry them through the water, and that's why you have the lipoprotein. So the lipoprotein's goal is to allow these fats to basically organize themselves in a way that they can be transported through the blood, which is water-based, to the tissues. So most of the LDL or HDL structures, these lipids, and then there's proteins on these lipids to help basically transport them through the blood, just so you can get an idea. And the lipoprotein, the apolipoprotein, is what has the binding sites, as we saw here, that allow it to interact directly with the proteoglycans in the vascular wall, and that'll become more important, particularly when we talk about lipoprotein little a. So now we're going to talk about immune cell inflammation. I want to get into it just a little bit more in depth than what we did before. So what we see is the recruited macrophages express several different polarization phenotypes and exert manifold effects in lesion development. Some macrophages attain a pro-inflammatory M1-like phenotype, possibly through binding of modified LDL to pattern recognition receptors, e.g. toll-like receptors, and they secrete pro-inflammatory cytokines, interleukin-1 beta, and tumor necrosis factor alpha, enzymes, and reactive oxygen species that promote further retention and modifications of LDLs, e.g. myeloperoxidase, as well as many other mediators that have been shown to play a role in atherosclerosis, e.g. plasminogen activators, cathepsins, and matrix metalloproteinases. So what we're seeing here is that when the monocytes, so when the white blood cells are in the blood, they're called monocytes. When they migrate to the tissues, they become macrophages. Macrophages, kind of think of them like Pac-Man. They can like kind of gobble up inflammatory components or cell debris or bacteria, etc. And so what happens is when these macrophages interact with LDL, particularly LDL that is oxidized, it triggers these immune responses through toll-like receptors. And the toll-like receptors are essentially, they're kind of like, I guess let's call them metal detectors for the immune cells. And when the immune cells wave these metal detectors over certain components, they trigger this response saying, hey, this individual has a weapon or whatever it is, or this component is not good. This component is problematic. So oxidized cholesterol or lipids are problematic. And so that triggers the monocytes, macrophages, to say, hey, we have a problem here. And then they start secreting pro-inflammatory cytokines or signaling molecules. And those signaling molecules attract other immune cells and have a whole host of functions, including producing reactive oxygen species, which is problematic in the vicinity of lipids, because those lipids can then further become oxidized. So when that happens, you get more, it's kind of like a feed-forward reaction where those lipids become oxidized and macrophages then upregulate more immune response, and you build more and more and more fats or oxidized fats in underneath the endothelial layer here in this new neointimal layer. So that's a huge problem. Something to mention here again is that if your LDL is rich in polyunsaturated fat, particularly omega-6s, this process is going to be amplified. Essentially, that omega-6 is kind of like dynamite sitting inside the LDL particles and when exposed to reactive oxygen species and other oxidizing components, essentially will more likely oxidize and trigger that oxidation of the cholesterol as well. So again, probably better or probably best to not have high loads of omega-6 within the body. Ideally, you'd want to see more monounsaturated fats or saturated fats inside the lipoproteins than polyunsaturated fats. And again, this image is basically showing you have the monocytes moving across the vascular wall, becoming macrophages, interacting with the fatty acids, forming foam cells. We'll talk about that in just a few minutes. Something else that's interesting here is you have the vascular smooth muscle cells differentiating into these macrophage-like cells as well. So they become like these immune-like cells. And then they can also gobble up or eat up in a Pac-Man-like fashion some of these damaged lipids inside the vascular wall. So let's go to the next one. So this whole process we're going to go through as we go forward is like a step-by-step process of this plaque formation with some microscopic images and then some more in-depth graphics than the other one before, so you guys can kind of see. So basically, with the immune cells, the macrophages that we just talked about turn into something called foam cells. And essentially what foam cells are these obese immune cells that are just filled with fatty acids. They eventually die from the toxicity of these oxidized lipids, but you can kind of think of these as like obese immune cells. The image that always comes to mind for me is like the people floating around in that movie WALL-E. It's like those are the immune cells now. Like they're just bloated and they can't move really well and they can't function really well. So what you're seeing here is the quote we see is both macrophages and dendritic cells serve as deposits of lipids from the insidating. So insidating means this movement of the lipids into the vessel wall. So the insidating lipoproteins, and become foam cells by mechanisms that remain incompletely understood in vivo. It may involve scavenger receptor mediated uptake of oxidized LDL, hydrolysis of free cholesterol from aggregated LDL complexes in extracellular lytic compartments, direct uptake of native LDL or remnant lipoproteins, or indeed a combination of these or other proposed pathways. So essentially, by taking up these lipids, they don't know exactly what allows the immune cells to become foam cells, but essentially they just take up a bunch of these damaged lipids and just become engorged with these lipids, and they sit inside the in the neointimal layer underneath the endothelial cells. So Chris Masterjohn actually has a really great video series discussing some of this. And basically what you can see is that oxidized lipids, oxidized LDL, directly damage the endothelial cells lining the vessels. So the hypothesis that Chris proposed there was that the oxidized LDL is gobbled up by the immune cells and moved into the vascular wall here to kind of protect the endothelial cells that are lining the vessel to some extent. So it's almost a protective mechanism. Again, from this perspective and from the perspective I like to look at things, I don't see things in the body as random. I see as there's a purpose. So I think that sounds like a pretty decent hypothesis overall. And the other thing to talk about is Linus Pauling's hypothesis is that the lipoproteins moving into the vessel wall are a way to kind of spackle over damaged areas of the vessel wall in subclinical vitamin C deficiency. So you don't have full-blown scurvy, which is actual vitamin C deficiency. You have like a subclinical level, so you're able to function appropriately, but you slowly develop vascular damage over time in the with subsequent plaque formation because you don't have adequate vitamin C to maintain the structure of the vessels. So I like both hypotheses. I don't think you have to choose one or the other. I think they both have a place here, considering the mechanisms, and it's a multifactorial process that has many different components working simultaneously. So I don't think it's just going to be like one thing solves it. That's why I wanted to put this whole picture together, and we can see that excess sympathetic activation and renin-angiotensin aldosterone activation leads to vascular damage when things like endothelin 1, and then you also have this lowered thyroid function can impair vascular function and increase the damage from the RAS system to some extent, and then you have the damage from polyunsaturated fatty acids, inflammatory responses like inflammatory stimulators like endotoxin can lead to vascular damage, or excess oxidative stress from iron can worsen the process as well. So that's why there's multiple factors. So it's not just about addressing one, like high cholesterol. It's kind of that's like only one thing to focus on here when there's so many other things going on. It gets so much airplay, but I feel like considering the whole process, when you kind of break it down, it's like that is one of the least important things to focus on. The question is more of like, why are you having this vascular damage? How can you repair that vascular damage, and what is triggering an inflammatory response and elevating the lipoproteins, etc. So those are more the questions I have, instead of like, oh, your cholesterol's high, let's just lower it. So again, looking at the process really brings this to light. And again, so what we're seeing here in the pictures, we're seeing the formation of the building up of these foam cells, and in the microscopic image here, you're seeing the foam cells as well underneath in this neointimal layer underneath the endothelial cells. And a xanthoma here is just kind of like a fatty buildup, and we'll talk about that as we go. So here, the next thing they're talking about is the source of plaque fatty acids. So what they say is the chemical composition of the necrotic core indicates that other sources of lipids may be important co-contributors, including direct accumulation of cholesterol esters from insudating LDL and free cholesterol-rich erythrocyte membranes deriving from intraplaque hemorrhages. It's also possible that macrophage catalyzed extracellular hydrolysis of aggregated LDL contribute to the high content of free cholesterol in the necrotic core. So they call this the necrotic core. You can kind of see it here. And essentially necrotic in the sense that there's no cells there. It's literally just these pools of fatty acids, these lipid pools that are not, there's no cellular material. And I kind of tried to show you guys this is like these little white pockets here, and then this kind of like darker bluish pockets here. There's no cells there in that arterial wall. It's just a buildup of these fats, these oxidized, damaged fats. And what they're saying is that these fats are coming from the lipoproteins, and they're also coming from what you can have in the artery is this angiogenesis, so this new vessel formation to feed the cells within this arterial layer. And that new vessel formation can lead to like the flow of red blood cells into this arterial layer or into the plaque, and we'll see that in just a minute. And essentially, the membranes of red blood cells, erythrocytes, can also contribute to this to the lipids that are present here. So it's really important to understand what type of lipids you have present for the cell membrane of the red blood cells, and then what's your lipid composition in your cholesterol. Those things are quite important. Even more so, at least to this atherosclerosis process, than what you have stored necessarily inside your tissues, because the fatty acids stored in your tissues, they're kind of locked away to some extent. Now, if you're under, and this is something that Pete talks about, if you're under stress, those fatty acids can become liberated. So if your tissues are very high in linoleic acid, when you're under stress, fatty acid lipase can basically break down those fats and then transport them out from the fat tissue, and then you can have higher circulating polyunsaturated fats even if you're not eating them. So that's why it's important to avoid storing large amounts of polyunsaturated fatty acids in your tissues over time, not to mention that they also have other effects on the tissues, and as far as becoming inflammatory mediators or being the precursor to inflammatory mediators, or altering membrane composition. And again, membrane composition can be quite standard, although drastic changes in diet can change the composition to some extent in the cell membrane. We'll talk about that in a completely different video, and like talking about the membrane pacemaker theory of aging. So I do plan on covering that, just not right now. Next thing they talk about here is the reversible fatty streak in the arterial wall. So you can develop a fatty streak in the arterial wall here, and it doesn't necessarily mean there's a problem. It can be reversed, and we're going to read the quote in a second. And that's just the fatty buildup in the vessel. However, once you start getting tons of like immune cell infiltration and whatnot, the process becomes, I think, a little bit harder to reverse. But what they basically say, foam cells, easily recognized by light microscopy, are telltales of lipoprotein-driven inflammation occurring in the vascular wall. They initially accrue within the proteoglycan layer of the intima. We discussed those proteoglycans building up and then trapping the LDL particles in the intima. And when several layers have formed, they are visible to the unaided eye as yellow-colored xanthomas or fatty streaks. We can see that here. Xanthomas are harmless and fully reversible if the stimuli that cause their formation dissipate. They are present already in some fetal aortas and infants in the first 6 months of life, probably reflecting risk factors of the mother, but their number decline in subsequent years. At adolescence, they reappear in atherosclerosis-prone regions of the coronary arteries and the aorta in most people. Okay, so basically what you're showing is that this process is reversible, assuming that the initiating factors are decreased and the body is able to heal these things. So that's when we talk about decreasing the amount of PUFON cholesterol so you don't get this oxidized cholesterol, decreasing inflammatory stimuli. It could be gut-derived endotoxin. It could be iron overload. It could be smoking is another huge one. It could be like heavy metal exposure. It could be subchronic or subclinical vitamin C deficiency. And now the subclinical vitamin C deficiency doesn't necessarily damage the vessels directly. It just makes it harder to repair the vessels, and we'll talk about that in another video. So it's kind of like when I look at heart disease, the things that I'm looking at are how can I minimize the damage, the sympathetic nervous system, renin-angiotensin, aldosterone system, excess glucocorticoids, PUFA, endotoxemia, inflammatory stimuli, excess iron. Can I minimize all of those? And then can I improve the ability of the vasculature to heal itself? Am I providing adequate vitamin C? Am I providing adequate amino acids in the like collagenous amino acids to build this collagenous structure? Am I providing adequate vitamin E and other components, maybe olive leaf extract, to minimize LDL oxidation? Am I minimizing my polyunsaturated fatty acid intake and incorporating more monounsaturated fat in my diet? All of these, or am I eating appropriately to optimize my microbiome to minimize that low-grade endotoxin transfer from the gut? Those are all things that I'm thinking about when I see cardiovascular disease, not just, oh, high cholesterol, let's lower it. Another thing is, if you do have high cholesterol, could that having a higher amount of cholesterol make this process worse? Yes, it can make this process worse because you have more fodder for the fire. However, the question is really like, why is that cholesterol elevated in the first place? Is there some inflammatory stimuli leading to that? Is there some metabolic aberration, aka the people that you see in, or the people that I've worked with and seen from super high fat, low carb, carnivore type diets with really high cholesterols, these hyper responders, probably not good in the long run. And then how do you lower that? So like one way to lower that is make sure your thyroid function is adequate. That'll increase the LDL receptors on the cell and then increase the amount of cholesterol that is moved into the mitochondria and converted to steroid hormones. So thyroid hormone directly lowers serum cholesterol by pushing that cholesterol into the cells and pushing that cholesterol towards steroid hormone production. So that's where you want the cholesterol to go, not kind of floating around in large amounts in the bloodstream, unless you're under stress or an infection, in which case the cholesterol is protective. But again, the goal would be to minimize that stress and eliminate the infection, because these processes shouldn't be running on chronically. So that's kind of tangential. We'll continue the next slide here. So now we're talking about the necrotic core formation. So what they say is many xanthomas do not progress further, but some, especially those occurring at predilection sites. We talked about those predilection sites: aorta, coronary arteries, the carotid arteries, large femoral vessels. Develop into progressive atherosclerotic lesions. The defining characteristic is the accumulation of acellular lipid-rich material in the intima. I discussed this before. Smaller lipid pools are initially seen beneath the layers of foam cells without gross disruption of the normal structure of the intima. In some lesions, the isolated lipid pools grow into confluent necrotic cores, or synonymously lipid-rich cores, through the invasion by macrophages. This process irreversibly disrupts the normal structure of the intima and leaves behind a matrix-devoid gruel of lipids and cell debris. So this necrotic core you're seeing here and you're seeing over here is literally just like these fatty acids with no cellular component, no living cells there, just like dead cells kind of hanging out. And this is like more advanced progression of the disease process, and this starts to disrupt the actual arterial structure. So initially, when we're just at this fatty streak level, the artery isn't, like, the gross structure of the artery and function isn't super impaired. You just have some fat in this intimal layer. So it's not ideal, but it's also like it can be reversed. But when you start getting to these like later stages, you start to see the breakdown of the arterial structure and these kind of like dead spots in the artery of just these nasty oxidized lipids and dead cellular components. And they call those that necrotic core. Now what we're going to talk about is, I think the next one's angiogenesis. So there we go. New vessel formation into the plaque. So that's what angiogenesis is. Well, I'm going to read the quote here. So neovessels, mainly originating from the adventitial vasa vincorum. So remember, the adventitious, that outer layer of the artery, the connective tissue layer, grow into the base of progressive atherosclerotic lesions and provide an alternative entry pathway for monocytes and immune cells of unknown quantitative importance. The plaque neovessels lack supporting cells and are fragile and leaky, giving rise to local extravasation of plasma proteins in the erythrocytes. Such intraplaque bleedings are common in fibroatheromas and may expand the necrotic core and promote inflammation. So these vessels move, basically all this dead tissue and whatnot here, and all these inflammatory signaling mediators trigger the vessels to actually, new vessels to actually grow into the plaque to kind of bring more immune cells and kind of solve the problem. The problem is that the new vessels are kind of leaky, so you get these red blood cells leaking there, and now you have heme iron inside this necrotic area that can oxidize more lipids, and you have these inflammatory components and more immune cells coming in. So it kind of just like makes this process even worse. This gnarly wound on the vessel that was kind of formed a scar a little bit, kind of thickened up a little bit. Now it's like growing, becoming more swollen, becoming more problematic. And then the new vessel formation is a huge problem. Something to keep in mind is this angiogenesis process is a process that you see in wound healing as well. And it's helpful in normal situations of wound healing. However, with this chronic inflammatory stain and damage to the vessel, it could possibly make things worse due to the dumping of these red blood cells and new immune cells into here if this is not getting resolved. So it can kind of feed into the reaction, although the angiogenesis process is a normal process in healing to provide more growth or more oxygen and nutrients to the damaged tissues. So it kind of can be a double-edged sword, and here we see some of the negative effects with this. Also, angiogenesis, something it is heavily involved in tumor growth. The tumors can upregulate those pathways and grow more vessels to them to extract more nutrients. But that's kind of neither here nor there for this topic. So again, angiogenesis can be beneficial or it can be problematic depending on the context. But it does indeed have a purpose. So now, next thing, fibrosis. So what you see in all kind of wounds in the body, particularly wounds that aren't healing well, is you have damage to the tissue, inflammatory response. After the inflammatory response, you get, if the healing isn't done appropriately, you get a fibrotic response. And then after the fibrotic response, you get calcification. So we have the inflammatory response we talked about. Now we're seeing the fibrosis. So what the quote shows us here is the connective tissue of lesions is initially that of normal arterial intima or adaptive arterial intimal thickening. But gradually, this loose fibrocellular tissue is replaced and expanded by collagen-rich fibrous tissue, which often grows to become the quantitatively dominant component of plaques, tissue that lie intertwined between a necrotic core and toward the luminal surface of the plaque fibrous cap are fibrous with a high content of type 1 collagen. So the plaque, when all this tissue dies and you have this necrotic core and you have these cellular components and you have this massive inflammatory response in this area, the artery and the cells, just instead of having these working normal arterial intimal layer, like these normal cells and the normal function that you would have there, things just become this massive scar, this fibrotic tissue that's largely non-functional. And you can see here that this you have the necrotic core here with the vessel moving in and all this dead kind of tissue or dead cell components and oxidized fats here, and you get this thin layer of fibrotic tissue that kind of walls it off. Now this will become problematic as we see in a bit, because walling it off with this thin fibrous layer, if this wall breaks, well, now you have an embolism. Now you have the formation of a component of this plaque component that can travel somewhere else and maybe possibly block another artery. And then also, when this breaks here, you have like kind of an inflammatory response that can kind of close off this local arterial area as well. So last piece we talked about. So I talked about inflammation, and then I talked about fibrosis, and then you see calcification. So what we see here is calcifications are common in progressive atherosclerotic lesions and increase with age. Apoptotic cells, extracellular matrix, and necrotic core material may act as a nidus for microscopic calcium granules, which can subsequently expand to form larger lumps and plates of calcium deposits. The necrotic core can completely calcify with time, and calcifications can constitute most of the plaque volume. So as the plaque becomes more fibrotic, the calcium granules and calcification can occur in these areas. Now, this is where parathyroid hormone can come into play. So strong elevations or this chronic elevations of parathyroid hormone can lead to these higher serum calcium levels. And the calcium that's coming from that parathyroid hormone is coming from the breakdown of your bone tissue. So it's breaking down your bones and then releasing calcium into the bloodstream. And then that calcium, if you have damaged vessels and you have these fibrotic areas, can capture the calcium granules and then calcify these tissues, which isn't ideal. And this is where looking at a coronary artery calcium score can become important, because it's basically showing these ladder processes of plaque formation where the calcium is deposited inside these damaged tissue areas. So the note on the parathyroid hormone is adequate vitamin D, A, K, calcium, and magnesium can help to suppress parathyroid hormone, provide adequate calcium, and not have these massive increases in serum calcium, and while preserving bone tissue. So a lot of clients will ask me, oh, isn't calcium going to cause my vessels to calcify? It's like, not necessarily. If your vessels are healthy and functioning well, they'll just distribute the calcium to where it needs to go, particularly to bones. If you have damage to your vessels, sure, maybe you have some calcium move there, but even if you don't have adequate calcium intake, your parathyroid hormone levels will increase, and then now you're increasing your serum calcium from the parathyroid hormone at the expense of your bones anyway. So I think calcium overall is helpful. Now, something to keep in mind is that massive doses of calcium in one sitting can drastically spike serum calcium as well, and then that can increase any deposition if you have vascular inflammation. But if you keep your calcium doses, I think if I remember correctly in the studies, under 300 milligrams in one sitting, so maybe 250 milligrams in a sitting with a meal, you probably won't get massive spikes in serum calcium, but you may be able to decrease your parathyroid hormone levels, especially if you combine with magnesium, vitamin A, vitamin D, vitamin K, etc. Something else to keep in mind here too is that parathyroid hormone is directly inflammatory to the vessels as well, especially in higher concentrations. So you're going to want to keep that low in this process of cardiovascular disease. Okay, so this is the last slide here that we're going to go over, and what this slide is showing us, and I'll read the quote in just a second, is that even when the arteries are heavily blocked or heavily damaged, they still maintain a lumen size that allows for blood flow. And the way they're allowing for that blood flow is instead of closing off the lumen, they just kind of expand outward. So the quote here says, during atherogenesis, the local vessel segment tends to remodel in such a way that the lumen area is usually not compromised until plaques are large, expansive remodeling. So expansive remodeling here means that the plaque is actually expanding out instead of narrowing the lumen of the vessel. Thereafter, stenosis formation may occur through continued plaque growth or shrinkage of the local vessel segment, constrictive remodeling, or a combination of the two processes. Expansive remodeling is more often seen with fibroatheromas, and the extent of enlargement is positively correlated to plaque inflammation, medial atrophy, and the size of the necrotic core. So as the intimal layer, the neo-intimal layer increases, it expands outward, and as there's more inflammation and more necrotic core formation, that vessel expands outward. So what we're seeing here is that at 0 to 50% of like vessel or formation of a vessel atheroma, so they're saying percent cross section, what you're seeing is that the vessel can still maintain a lumen of about 4 millimeters. So the same as if the vessel didn't have any formation of the atheroma or the plaque. However, and that's because the vessel, again, is expanding outward. However, as the vessel gets to like 75 to 89% vessel occlusion, or 75 to 89% plaque formation, you see that lumen start to narrow. So here we see it's at 2 millimeters. Now, despite when you're getting to 75 to 90%, again, you're still only at 2 millimeters. What you're seeing in terms of vessel lumen diameter, what you're really seeing though is that the other areas of the vessel, like that neo-intimal layer, expands outward drastically. So the vessel kind of becomes bloated, but still kind of tries to keep the lumen. And this is important because you still need blood to flow. Again, as I talked about in the beginning, if you don't have blood flow to the tissues, you don't have oxygen, you don't have glucose, you don't have fatty acids, you don't have vitamins, you don't have minerals. So the cells don't have the raw materials to maintain general function. They just die. And you see that very quickly with a heart attack. If the coronary artery vessel is blocked off for any extended period of time, a couple minutes, the tissue on the heart starts to die because it can't keep up with its energetic demand because it doesn't have oxygen and it doesn't have nutrient. And so eventually you damage the energy producing ability of those cells. Those cells eventually die. And you get formations of like fibrosis and necrosis on different areas of the heart that were supplied by that vessel. So the vessel's really trying to stay open. The body is trying to keep those vessels open over time, even if they're under like stress and damage. It's trying to keep that lumen open to supply that blood. So that's basically it for this video today. Hope you guys enjoyed this, going through the different mechanisms of the formation of plaques within arteries. I'm gonna talk about some other factors like iron, endotoxin, the renin-angiotensin-aldosterone system, maybe endothelin-1 with that. We'll talk about thyroid. We'll talk about the sympathetic nervous system and glucocorticoids, vitamin C, polyunsaturated fats, and then iron. So there's quite a few things. And again, that's why I titled this, I think it was coronary artery disease and friends, because there's so many different extra components. So we'll get into that in subsequent videos. Hope you guys like the content. I'll catch you in the next one.